Hello, I'm Jane Mangold from the Agricultural Research Service and the Eastern Oregon Ag Research Center, here to present today's Ag Minute. At the Ag Research Station, we seed many test plots in rangeland degraded by juniper expansion and other weedy species in an attempt to restore more desirable plants. Although many factors beyond our control will affect the success or failure of those seedings, the choice of seed material is one factor we can control relatively easily. Whether you are choosing seeds for a large-scale rangeland restoration project, a medium-scale pasture revegetation, or a small-scale landscaping renovation, choosing quality seed is important to ensure the best possible results. This week I will be discussing how a species is selected and increased for commercial seed sales, seed testing requirements, and seed labels. All seeds are not created equally, and throughout the week I'll help you determine how to choose the highest quality seed for your next seeding project. I'm Jane Mangold. Please tune in tomorrow when I'll talk about developing seeds for commercial use. Acquisition of quality seed in the quantity needed is essential for successful restoration or revegetation projects. After the need for a species or suite of species is identified, a seed increase program begins. For example, many grasses native to Europe and Asia, like crested wheatgrass and smooth brome, were developed in the early to mid-1900s for revegetating severely degraded rangeland in the western U.S. Researchers collect seed from wild populations of the species they are interested in and take the seed back to their research facilities. After cleaning the collected seed, they put the seed into seeding trials in the environment they wish for it to grow. Plant performance in the trials is evaluated for 2 to 3 years. Researchers then select the best populations for increase in cultivated fields based on characteristics like plant height, vigor, biomass, and seed production. This seed goes on to become foundation seed, the seed which is eventually distributed to growers for commercial production. It may take 5 to 6 years from initial identification of the need to commercial availability of the seed. Whew, that's quite a process. Kind of makes you appreciate that bag of seed a little more, doesn't it? Today's Ag Minute is about seed testing. Federal and state laws require that seed be officially tested and appropriately labeled so the consumer knows exactly what he or she is purchasing. Seeds are tested for purity, germination, dormant seed, viability, and other material including inert matter, other crop seed, and weed seed. Federal, state, and private seed testing laboratories in the U.S. and Canada are required to use standard testing procedures for most grasses used on rangeland. However, testing procedures for many shrubs and forbs, or broadleaf plants, have not been standardized, and laboratory test results may be inconsistent. If you are purchasing seed and the bag does not contain a label that lists test results along with the name and variety of the species, its origin and year of harvest, be leery of purchasing it. You may not get what you think you are paying for. Rather than buying your seed based on the pretty picture on the bag, you should turn to the somewhat mundane-looking seed label. The label contains important information about the quality of the seed. The kind and variety tells you the species and the variety if different varieties are available. Purity tells you what percentage of the material in the bag is seed of your desired species. Inert matter includes dust, stem, soil, chaff, and other non-seed items. Most grass seed should contain no more than 1 to 15% inert matter, or it will be difficult to plant. The number of weed seeds found in a given sample is also listed. Noxious weed seed is a separate listing on the label and includes seeds from plants considered noxious in the state where the seed was produced and tested. You should never purchase seed that has anything other than none printed next to noxious weed seed. Germination explains the percentage of seeds that germinated during testing. Origin, net weight, and date tested are fairly self-explanatory. The origin is important because it tells you where the seed was produced. Try to use seed that was produced as close as possible to the area you will be seeding. The label on a bag of seed makes comparisons between products much easier. A bag of seed contains matter other than your desired species, so it is important to know the amount of pure live seed, or PLS, in the bag before making a purchasing decision. To calculate pure live seed, multiply percent purity by percent germination, both of which are listed on the seed label. Forget Einstein's famous equation E equals mc squared, and instead remember PLS equals purity times germination. You may find that a more expensive but cleaner bag of seed is actually a better deal than a cheaper product that contains a large amount of inert material and weed seeds. And a product with higher PLS usually displays more vigorous, healthy seedlings. So, using cleaner seed will save you money, give you better results, and decrease the amount of weed seeds you are introducing to the area that needs to be revegetated. I'm Jane Mangold. Thank you for joining me this week.